Hello, everyone. I am Anisha Fartia, Product Manager at NanoString Technologies. Today, we have with us Maria Mattoprado, who is a graduate student researcher at the Imperial College London under Dr. Jonathan Krell and Adam Frampton. Today, she's going to discuss her research on microRNAs as bile-based biomarkers for biliary tract cancer using nCounter microRNA expression assay. Graduated at University of Barcelona as a biomedical scientist in 2013, she also holds a Master's of Science in Cancer Biology from the University of Kent. Her recent research is focused on discovering clinically useful microRNA biomarkers for pancreatic cancer. Without any further delays, I will hand over the webinar to Maria.
Good afternoon, everybody. Thank you for coming to this webinar today. In this talk, I will highlight the characteristics of pancreatic cancer, the importance of microRNAs in cancer, and I will show you some studies on biomicroRNAs. Moreover, I will present you some results from my study where we use NanoString technologies, particularly the nCounter microRNA expression assay, which validates the idea of using microRNAs as biomarkers for diagnosis of cancer. Pancreatic cancer is a lethal disease. It has a survival rate of 5%. This type of cancer has a poor prognosis because patients present non-specific symptoms, and this leads to a late detection. Pancreatic cancer is characterized by local invasion and early metastases, and the current imaging techniques are not accurate, sensitive, or specific, being the only effective treatment surgery. Although surgery is the therapy of choice, approximately only 20% of the patients benefit from it. Pancreatic cancer also accumulates genetic alterations, and it's difficult for a pathologist to differentiate tumors that are similar in morphology but different at the molecular level. Therefore, microRNAs in bile is becoming important. In this slide, I'm going to show you the process of microRNA. A microRNA gene is located inside the nucleus, and after a few cleavage steps, a pre-microRNA is exported into the cytoplasm. The pre-microRNA loses its hairpin loop structure and generates a microRNA duplex. Next, a protein formed by dicer cleaves the microRNA duplex, meaning the passenger strand is secreted and the leading strand binds to the argonal 2 protein, which forms the RIS complex. The RIS complex guides the microRNA toward its target, the messenger RNA. And at this point, two events could happen. The microRNA will be degraded or the translation will be repressed. Many studies have identified a panel of microRNAs that could discriminate healthy individuals from pancreatic cancer patients, and in turn, this panel might be used as a diagnostic biomarker in pancreatic cancer. However, just a few studies have been published until now that are based on biomicroRNAs. I would like to show you some of the published studies. In 2011, this was the first publication to confirm the existence of microRNAs in the bile fluid. These researchers found that microRNA 9 has a strong potential to be used as a clinical marker for biliary tract cancer. Later, in 2014, Lankish and co-authors found significant differences of microRNA levels in bile from patients that had biliary lesions after liver transplantation. And most recently, in 2016, this publication was the first to describe a polarized release of microRNAs from the liver into the bile during hepatic injury. And this was useful to distinguish biliary injury versus hepatocyte injury. After giving you a bit of background about pancreatic cancer, I would like to focus in our study. Our aim was to detect pancreatic cancer at an early stage, and to do that, we decided to use the biofluid because of the proximity to the malignant lesion and because it has been reported to be a rich source of biomarkers. Also, look for microRNAs because they are tissue and cell specific. Also, because of their stability and resistance to degradation. So, we start with 1 ml of bile that was centrifuged at low speed for 10 minutes and 40 degrees. Then we filter the submetan and we end up with half of the original volume, sometimes more, but generally around 600-500 microliters. Then we use twice LLS to isolate the RNA, and the RNA samples were shipped to NanoString for an nCounter microRNA panel. When we received the data back from NanoString, we decided to do some bioinformatic analysis in order to find which microRNAs were up and down regulated. We also used a software program called NormFinder to identify which microRNAs didn't change among all the samples. And we found 3, microRNA 188, microRNA 96, and microRNA 577. The panel we use is called nCounter microRNA Expression Assay, and it can detect up to 800 microRNA genes, which are selected based on previous publications of relevant biological data, and they are also available in miRNA. Particularly, this assay provides a direct detection and quantification of microRNAs in a single reaction without amplification. So going back to our results, we had 58 biosamples classified according to the diagnosis in benign, chronic PDAC, IPMN cancer, cholangiocarcinoma, and billary cancer. Benign and chronic pancreatitis are a subset of cohort samples for benign, and PDAC, IPMN cancer, cholangiocarcinoma, and billary cancer are the ones classified as malignant samples. This is a picture that shows the results from 3 bioinformatic analysis, and I would like to point out that only 1 microRNA is upregulated when comparing all cancers to benign cases. This is the case of microRNA 8 on the left side. Also, there are other microRNAs that were upregulated in cholangiocarcinoma samples compared to benign samples. For instance, microRNA B and microRNA H. From this list, we select a few of them to be tested. Here you can see the box plot for microRNA A. Below the black line are the benign cases, and above the line are the malignant cases. So, as I said before, we wanted to validate this data that we obtained from the nCounter microRNA Expression Assay, and to do that, we performed reverse transcription quantitative PCR. The bioinformatic analysis showed that microRNA A was upregulated in all cancers versus benign samples, and we used one of the 3 normalizers, in this case microRNA 188, and we agree that this microRNA is validated as upregulated in malignant bile cases compared to benign bile cases. However, it has a moderate discriminatory ability. On the contrary, when microRNA 577 was used as a normalizer, we still validate this microRNA as upregulated, and more importantly, the discriminatory ability improved up to 0.8. We also validate microRNA B, which is shown to be upregulated in cholangiocarcinoma compared to benign individuals. You can observe here the box plot for microRNA B. The green box is cholangiocarcinoma and is above the line, whereas the healthy individuals are below the line. Here, we can conclude that microRNA B was upregulated in cholangiocarcinoma in comparison to benign bile samples. However, this microRNA needs further testing as the size of the sample was small, because we only had 6 patients that present cholangiocarcinoma compared to the 17 benign patients. We obtained the same result when we used microRNA 577 as normalizer. Another microRNA that we tested was microRNA H. This microRNA was upregulated in malignant samples when compared to benign cases. And again, we validated as upregulated. However, although this microRNA has a poor discriminatory ability when microRNA 8 was used, its sensitivity and specificity improved when another normalizer, in this case microRNA 577, was used, as you can see in this slide. Here, there is another picture from the analysis, this time showing the upregulated microRNAs in PDAC. Again, from this list, we select a few of them to be validated. We focus especially on microRNA M. The microRNA M was upregulated in PDAC when compared to cholangiocarcinoma and to benign cases. Although the first analysis suggests that microRNA M was upregulated in PDAC, a further validation indicates that it wasn't upregulated, as you can see from the graph. However, when we use microRNA 577 as a normalizer, it validates the previous analysis, showing that microRNA M was actually upregulated in PDAC compared to benign samples. So probably one of the questions you might ask is why we chose these microRNAs. The selection of microRNAs to be tested or to be validated was based on previous bioinformatic analysis, and from that we obtained a list of approximately 20 microRNAs. We also looked into the literature and found some publications about a few of them. Then we decided to start the validation, selecting some candidates. And our conclusions from the study so far are that the class formed by microRNA B and microRNA C has been reported to be either deleted or downregulated in pancreatic cancer cells. And there is also the hypothesis that this cluster acts as a tumor suppressor. The restoration of this cluster leads to tumor regression, apoptosis, and inhibition of cell proliferation. Especially, we found that microRNA B was upregulated in cholangiocarcinoma patients. However, our bioinformatic analysis showed that it was downregulated in PDAC, which fits with the literature research and all the publications reported from this cluster. The other microRNA that we validated was microRNA M. It has been shown to be a negative regulator of p53, but the knockdown of this microRNA increases the p53 messenger RNA levels and promotes apoptosis. However, this microRNA has a controversy in our system, because when we use microRNA 188 as a normalizer, this microRNA was downregulated. However, when we use microRNA 577, we validate it as an upregulated microRNA. So it needs further validation, and it would be ideal to confirm which type of pancreatic cancer has this microRNA up or downregulated. And the last microRNA I would like to mention is microRNA 8. There is also controversy with this particular microRNA, because when we use two of our normalizers, we obtain totally different results. When microRNA 188 was used, it indicated that this microRNA was not upregulated, but microRNA 577 validates that upregulation. Moreover, other publications performed in vitro and in solid tumors and reported this microRNA production was inhibited in PDAC. One explanation I can think of is that the release of these microRNAs varies depending on the type of sample analyzed. And it's demonstrated that microRNAs from tumor tissues present different levels of microRNAs when compared to the levels of microRNAs in fluid. So the future work that has to be done is to validate other up and down regulated microRNAs, assess other possible normalizers, and the most important point is to validate these results in an independent cohort of biosamples from open surgery and preoperative ERCP. ERCP is just a procedure that examines the pancreatic and the bile duct. And we will try to use these microRNAs to identify cancer in preoperative bile samples before surgery to better assess the patient. Finally, I would like to thank my supervisors, Adam and John, and Professor Justin Stebbings, Action Against Cancer, which is a charity in the UK that not only funds my PhD, but also that is involved in other cancer projects. And obviously, NanoString for inviting me to this webinar. Thank you very much for your attention, and if you have any questions, I will be happy to answer any of them. Thank you.
Thank you, Maria, for an insightful presentation and sharing your research on microRNA in biliary tract cancer using NanoString microRNA expression assay from nCounter Technologies. So I do have a few questions over here. How do you normalize your data? What about spiking control?
Okay, our data was normalized with microRNAs that we identified using this software program that it's called nCounter. Basically, it's an algorithm that identifies the optimal normalization microRNA according to the expression stability among a set of candidates. And in this particular case, the nCounter microRNA expression assay profiled like around 800 microRNAs across our 58 RNA bio samples. And a spiking control like Zomia 39, which is this one, but as an exogenous control. And this exogenous control only shows you how good or bad is the RNA isolation or how good or bad is the performance of the RT-qPCR. So exogenous spiking controls are like quality control measurements. And in our study, we wanted to find an exogenous control that whose expression was strong and also didn't vary among all the samples. So that's why we used this software program called nCounter to normalize our data.
Perfect. Thank you so much. I do have another question here. How were or are the samples collected?
The samples were collected during open surgery and for a variety of benign and malignant biliary or pancreatic disorders. And the bile was collected from the gallbladder. And we wanted or we aimed to have a validation cohort from surgical patients, but also from patients from preoperatively undergoing endoscopic retrograde cholangiopancreatography, which is ERCP, as I said before. And in this way, we can translate our findings most effectively into improving patient care.
Sure, thank you. And just the last question, how accurate will these microRNAs be in order to distinguish PDAC from another type of pancreatic cancer? And I mean, the main concern over here is that the bile is not produced by the pancreas but the liver.
That's an interesting question. We believe that these microRNAs could be able to differentiate patients with pancreatic cancer from healthy individuals, but also that they could be used as a classifiers of different types of biliary disorders, like cholangiocarcinoma, chronic pancreatitis. It's true that the pancreas doesn't produce the bile fluid, but the liver does, and then it's stored in the gallbladder, and the gallbladder secretes that into the common bile duct. And the microRNAs are released from the pancreas into the pancreatic juice and can be found in the bile fluid. So this supports, in a way, our study.
Sure. Thank you so much for the webinar, and thank you so much for answering the questions. We really enjoyed hearing your presentation. Thank you once again.
Thank you.
So before we end our exciting webinar, I would also like to tell you about another exciting thing. So NanoString is offering a grant program, and we really want to help our researchers resolve the most complex problems in cancer. 4 applicants will be chosen by NanoString to receive an nCounter Vantage 3D assay of their choice from our menu of panels, and the award includes profiling of 36 reactions. The abstract submission deadline is January 31st, 2016. And to learn more, please visit our website at nanostring.com/vantage3D_grant. And you would be able to learn more about this grant program, as well as you'll be able to submit your grant submission over there. So we really look forward for your submissions, and happy researching. Thank you all for attending.